Hello, you are welcome back to Unit 4, Session 4. Under this session, we'll be looking at the mechanism of muscle contraction. And we'll begin with a description of contractile units called the sarcomeres. It is also important to have a deeper understanding on the relationship between the contractile proteins in the myofilaments in order to be able to explain the mechanism of muscle contraction. Hope you enjoyed this session also. By the end of this session, you should be able to define the term sarcomere and also name the bands and lines in the sarcomere and also explain why bands are light or dark, and finally explain the mechanism of muscle contraction. From the last session, we learned that the light and dark bands are due to the distribution of proteins in the myofilament. And the dark bands are also referred to as the A bands. And these A bands are named because they are anisotropic in polarized light, which implies that they vary in magnitude according to the direction of measurement by light waves. Under the light microscope, the parallel array of thick filaments are mainly responsible for the appearance of the A band. And in the center of the A band, there is a pale region, and this region is referred to as the H band, and H implies Hansen, and this can be seen in relaxed muscles. So this H band represents an area of thick filaments that are not overlapped by thin filaments. So at the middle of the A band or the H band is a line called the M line, and this is demonstrated in the figure in the next slide. As shown in figure 27, the dark band, which is also known as the A band, it has been indicated, and within the A band is the H band, and at the center of the H band or the A band is the M line. On the other hand, the light bands are also called I bands, and this is because they are isotropic in polarized light, which implies that they do not vary in magnitude according to the direction of measurement. And within the middle, or a line that runs through the middle of the I band, is the Z line. The cross striations of the myofibrils are serially repeating units, and this is referred to as the sarcomeres. And the sarcomeres is considered to be the basic unit of contraction in striated muscles. So a single sarcomere extends from one Z line to the next Z line, and it contains one A band separating two semi-I bands. And as shown in the diagram, thus each sarcomere contains two half I bands, one at each end. And because a single I band straddles the Z line, and because of that, it forms part of two adjacent sarcomeres. The myofibrils is composed of a series of the sarcomeres consisting of interdigitating thin filaments and thick filaments. The thin filaments are composed of the protein actin, and in addition are two important regulatory proteins, that's tropomyosin and troponin. The actin filament is said to be polar, and they contain a binding site to which myosin can bind. The thick filament, on the other hand, is made up of myosin protein, A molecule of myosin contains two heads and a tail, and the myosin molecule of the thick filaments are arranged with the heads showing or oriented towards each end and the tail towards the center, indicating that a bar zone that lacks head and contains only tails occur in the central region of each thick filament. The myosin heads function is that they act as the active sites for enzyme activity, and they are the site to which the actin protein also binds to. So in sarcomeres, the, it is arranged in the form that the thick filaments occupy the central portions of the sarcomere, whereas the thin filaments attach at each end of the Z lines and run parallel to and between the thick filaments. So far, we've learned that the I bands are made up of thin filaments, and each sarcomere has half of an I band at its ends, and the whole I band is shared between adjacent sarcomeres. The A band is also composed mostly of thick filaments and the thin filaments between them. Also, the H bands are composed of thick filaments, and in relaxed muscles, they represent the area between the ends of the thin filaments that are attached to each Z line at the other ends. In contracted muscle, when the thin filaments merge or overlap, the H band narrows or may disappear. As described in the previous slides, the sarcomere shortens during contraction, and the A band does not change in length. And because of that, the distance between the Z lines lessens, and the I and H bands also narrows. The sliding filament mechanism of muscle contraction helps in explaining how a muscle cell shortens on contraction. Looking at figure 28, in the sliding filament model of muscle contraction, the thin actin filaments attached to the Z line slide or they are pulled inward along the thick myosin filament, and the sarcomere, measured from one Z line to the next, is shortened. In muscle contraction, the laterally projecting hairs of the thick myosin filament comes in contact with the thin actin myofilaments and rotate on them, and this pulls the thin myofilaments towards the middle of the sarcomere past the thick myofilament. The Z lines also come closer together and the sarcomere becomes shorter. The length of the A band also remains constant and the length of the myofilaments do not change. So in summary, as contraction is occurring, the length of both the sarcomere and the I band decrease, but A band length remains the same. And as a consequence of the actin-myosin interaction, it leads to thin filament sliding into the A band, and this results in observing that the sarcomere shortens, the Z lines also pull closer together, the H bands narrow and disappear, and the I bands also shortens. The A bands remains the same, myofibrils and the muscles therefore shortens. The energy in muscle contraction is provided by ATP, that is adenosine triphosphate, and it results in the conversion of ATP into adenosine diphosphate, that is ADP, and inorganic phosphate. This results in the release of energy. And the enzyme that catalyzes this particular conversion is myosin ATP. And the reaction occurs in the presence of cofactors such as the calcium and magnesium ions. Under Unit 4, Section 4, We studied the sarcomere, we described it and got to know that it's the contractile unit of skeletal muscle. And we also further explained the basis of the banding and stretching of the fibers and the arrangement of the contractile proteins in the myofilament. And we ended our discussion with a description of the mechanism of contraction.